Welcome everybody to this presentation. My name is Silkman. The title of this presentation is Making a Genome. And today I'd like to discuss the science behind the Steve Avery and Brendan Dassey cases. Ken Kratz, during Stephen Avery's trial, made a very important statement. He stated, DNA kinds of evidence can't be confused. It can't be tricked on cross-examination. DNA evidence is what it is, and only through human failure to collect it properly, or failure to analyze it properly, or failure on my part to explain it properly to the jury is the limitation of DNA evidence. And note the next point he makes, because it actually is very critical. Because the DNA evidence in this case is incredibly powerful. So here we have Stephen Avery and Brendan Dassey. In Stephen Avery's case, there was DNA evidence, plenty of DNA evidence that was used against him. In Brendan Dassey's case, there was no DNA evidence. Both of them were sentenced to life in prison for the murder of Teresa Habach on the 31st of October 2005 at the Avery salvage yard. Ken Kratz stated, Mr. Avery achieved some degree of notoriety back in 2003 when he was exonerated for a 1985 sexual assault conviction. You should know that the exoneration was based upon DNA evidence. You should know that the DNA evidence was performed by the Wisconsin State Crime Laboratory, and it was performed by an analyst, the head of the DNA unit in Madison, a woman by the name of Culhane. I want you to remember that name because you're going to hear that name later on in this case. And this is Culhane. The importance of Cherie, of course, is that she carried out all of the forensic DNA evidence that was held predominantly against Stephen Avery. But we need to ask, what actually is the basic structure of DNA so we can get to understand this case at a finer level? If we look at a DNA molecule, essentially DNA is double-stranded and it forms a helical structure, and it's known as the DNA double helix. The DNA strands contains bases. There are 4 bases, A, G, C, and T. And specifically, A base pairs with T, G base pairs with C. Now, this information is critical because all of the molecular biology tests rely on this fact of base pairing. So here is a molecular structure of DNA. The question we want to ask now is exactly where do you find DNA in a human cell that is suitable for forensic analysis? Here is a typical cell, a typical eukaryote cell, a human cell. And what you note is there is a nucleus present. There are also these structures within the cell known as mitochondria. These are the 2 predominant sites in a human cell that contain DNA. But it's important to note that there is a big difference between the 2 DNA molecules. In the nucleus, it contains so-called genomic DNA. This DNA is present on chromosomes. And there are 2 chromosomes or 2 copies of each chromosome per cell. Mitochondria contain mitochondrial DNA. Note, the important thing here is that the mitochondria contain multiple copies of mitochondrial DNA per mitochondria. Both genomic and mitochondrial DNA can be used for forensic DNA analysis. So the important point here, guys, is that both the nucleus and the mitochondria contain DNA. If we have a look at the DNA from the nucleus of a cell, we can see that it is present in a structure called a chromosome. This chromosome contains packaged DNA. Scientists are able to isolate or remove the proteins from a chromosome, and you get this structure here. So all the loops that you see here and shown in a black arrow, this represents one massive DNA molecule which is present in one chromosome. So genomic DNA is extremely long. And here's the problem. If you expose DNA to very high temperatures, such as those you'll find in a fire, you will destroy the DNA molecules. In essence, the DNA will be essentially useless for forensic DNA analysis. Now, we have, as I stated before, inside our cells, structures called mitochondria. They are also known as organelles. Inside the mitochondria, they contain circular mitochondrial DNA. Each mitochondria is inherited from the mother. Therefore, all your mitochondria are inherited from your mother. That's important. So, why is this good? Well, a typical cell has around about 100 mitochondria. Each mitochondria contains about 5 circular mitochondrial DNA molecules. That means that one cell contains about 500 copies of mitochondrial DNA. That's important, especially for forensic analysis. So, mitochondrial DNA is ideal for forensic DNA analysis. Why is that? The mitochondrial DNA is much smaller in size, and it's likely to survive intact following a burning episode, especially during high exposure to high temperatures. So, as a forensic scientist at a crime scene, you want to know where you can obtain DNA sources for testing. Essentially, any cell in the body that contains a nucleus and mitochondria is suitable for the extraction of DNA. For example, DNA can be isolated from white blood cells, and these are normally present in a blood sample. Epithelial cells, which are present in saliva, and they can be very easily obtained by use of a buccal swab. Skin cells and tissue, like muscle, are a rich source of DNA. Also, hairs that contain a follicle bulb, you can isolate DNA. And I just want you to note that this was what was used in order to exonerate Stephen Avery. Teeth and bones, including those that have been burnt in a fire, will have sufficient DNA left over to isolate the DNA from. Now remember, we talked about the mitochondrial DNA. It is highly likely that some mitochondrial DNA will be able to be extracted from burnt bones. Urine and semen are other sources where DNA can be obtained from. And as you can see in the diagrams below, there are multiple cells in our body in which DNA can be extracted from. Here's the big question. How does one actually obtain a DNA profile? The first thing that must be done is that the DNA must be extracted from so-called test samples. A forensic scientist will normally extract DNA from blood or a buccal swab. So if someone takes a cheek swab of your mouth, that'll have enough cells in it to extract DNA from. The DNA is then subject to DNA amplification. You can buy special kits, and Cherie Culhane in her laboratory used the Promega PowerFlex 16 kit. This contains all the components in order to amplify up the DNA. A special process called capillary electrophoresis was used, and this allows a DNA profile to be obtained from the DNA that's been amplified. Note, special markers are used. They're called short tandem repeat markers. And in total, 15 markers were used in order to get a DNA profile, plus a gender marker. The gender marker allows the analyst to determine whether the sample is either from a male or from a female. Once a DNA profile is obtained, the sample can be compared with the CODIS system. It's basically a DNA database, and it's kept by the FBI. And the question you can ask is, do I get a hit or a match with someone in the CODIS database? This is precisely what Cherie Culhane did when she did her initial testing on some DNA samples, and Stephen Avery's name was able to be matched in the CODIS database. The other thing you can do is compare the DNA profile to other DNA profiles found at a crime scene. And you could ask the question, do I get a match or not? Now, the short tandem repeats are extremely powerful molecules to use. The short tandem repeat represents a specific region of DNA, and that's called a locus. That region of DNA resides on a human chromosome. A specific locus has alleles, and all that means is that the alleles are of different sizes, and this becomes very important when you're trying to derive a specific DNA profile. Now, essentially, you inherit one allele from each of your parents, and I will go through the exercise to see if you could understand it. Importantly, you can either be homozygous, that is, you have two identical sized alleles, or heterozygous, in which the alleles are of different sizes. Let's have a look at an example. Now remember, what I've done is I've simplified it to explain the process of inheritance. So say we look at a mother, and a mother, of course, has got two specific chromosomes which are identical, and on that chromosome, or in this case a DNA molecule, we have a specific locus, locus A. That locus happens to be 110 bases in size. On the other chromosome, there also is locus A, and it's slightly different. It's 140 bases in length. The father, on the other hand, again has two identical chromosomes, and on one chromosome has a locus A of 110 bases, and on the other chromosome has a locus A of 180 bases. In the offspring, when the egg meets the sperm to produce an offspring, that offspring can inherit this particular piece of DNA from the mother. And notice it contains locus A of 110 bases. The other chromosome that it inherits from the father just happens to be this one here. And that locus A also is 110 bases. So the offspring contains two chromosomes, two specific chromosomes, each with a locus A of 110 bases. That means that individual at locus A is said to be homozygous. Now, if you've noticed, we have alleles. That means if you look at locus A, it can come in a variety of sizes: 110 bases, 140 bases, and 180 bases. These are known as alleles. So STRs are critical, because what you're trying to do with an STR is to develop a unique profile of an individual. Now, this is someone who is homozygous. Let's have a look at the alternative. And that is the mother, as we've had previously, has got a locus A of 110 bases and a locus A of 140. The father has a locus A of 110 and a locus A of 180. In this case, the offspring inherits locus A of 140 bases and locus A of 180 bases. So this individual is now said to be heterozygous. In other words, both locuses are different. Okay, so the machine produces a DNA profile, and let's determine how we can interpret this DNA profile. If you have a look at the CSF1PO locus, one can see that it contains one unique band that appears. That means that this individual has two copies which are absolutely identical. So for the CSF1PO locus, that individual is homozygous. For this particular locus, the D2S1338 locus, you can see two distinct peaks. That individual is heterozygous. So each person that is tested should have a unique DNA profile. So now you can understand the power of generating a DNA profile of an individual, especially at a crime scene. It can either include you as being present in the crime scene, or it can exclude you. That's very, very powerful evidence. Now, I've only really skipped over the DNA STR typing. If you do want to know a little bit more detail of this, there's an excellent YouTube clip, and I invite you to have a look at your leisure, which will explain it in more detail. Now, let's have a look at Stephen Avery's trial. And let's have a look what Sherry Culhane found during her testing. She found Stephen Avery's DNA profile in several locations inside Teresa's Toyota RAV4. It was also found on the hood latch of Teresa's RAV4, and it was found on Teresa's spare key, and that key was found in his bedroom. So I'm just presenting the findings. We can spend several hours easily discussing the controversies of these results. Now, what typically happens is that a report is written up, and this shows Exhibit 311. So they had the RAF4 in the laboratory, and what Cherie Culhane did was swab various parts of the RAF4 and was looking in particular for stains, that is, blood stains in the RAF4. So here's an example of item A1 and item A8. But remember, in the end, all of these samples were tested. At the same time that they were swabbing the RAV4, they also had Stephen Avery's Pontiac Grand Am in the laboratory, and they were swabbing the inside of that vehicle as well. Again, here's an example of item B1, which was a stain found inside the Pontiac Grand Am. And finally, just to show you, they did a swab of item C, which is the Toyota key that was found in Stephen Avery's bedroom. So the results are shown here. The DNA profile results are shown here, and you can see quite clearly that Teresa Habach's DNA as blood samples was found in the rare cargo area of the Toyota RAV4. For example, sample A1. Her DNA was also found on a wild cherry Pepsi can, shown here as item A14, and that was derived from saliva. Now some of you might be thinking, well, wait a minute, where did they get a profile of Teresa Habach if her body was essentially cremated? And the answer is they obtained her DNA profile from a pap smear that was done several years ago. Both the state and the defense had a gentleman's agreement that no one was going to contest that pap smear had indeed come from Teresa Habach. Let's have a look at the DNA profile results regarding Stephen Avery. So here is the full profile of Stephen Avery derived from a buccal swab. And these are some of the items that were tested. Item A8, which was a stain near the ignition key. Item C was the Toyota key. And item B1 was isolated from the front console of Stephen's car. You can see that it's a 100% match to Stephen Avery's profile. This means, of course, that Stephen Avery's DNA was found inside Teresa's RAV4 and also on the key. It also, if you remember, was found under the hood, on the hood latch. But there are other items of equal importance. Item FL. Item FL was a bullet fragment, and it was found in Stephen Avery's garage on March the 2nd, 2006. Now remember, his garage had already been extensively searched by many officers. Suddenly, on March the 2nd, they find this bullet fragment. They also found another one. This bullet fragment, item FL, was only found after Wiegert, during an interrogation session, mentioned to Brendan Dassey who shot her in the head, and this was done on March the 1st, 2006. Cherie Culhane did a DNA profile, and on that bullet they found Teresa Habach's DNA. But note, no blood or tissue was seen on the bullet, on item FL. And I want to be upfront and just say that the DNA analysis done by Culhane was extremely controversial, especially for item FL. But it was a crucial piece of evidence for the state, because now they had the narrative that Brendan Dassey, after being told by Wiegert about who shot her in the head, Brendan Dassey stated that Stephen Avery did. Lo and behold, we find a bullet, and it just so happens to have Teresa Habach's DNA on it. There's another controversial item, and that's known as item BZ. Bones were collected from very heavily fragmented and burnt bones were collected from 3 separate locations. One, Stephen Avery's so-called burn pit, the Dassey burn barrel, and the Manitowoc gravel pit. A small piece of tissue was able to be removed from the heavily charred piece of bone known as item BZ. And note, only a partial DNA profile that matched to Teresa was able to be obtained from item BZ. Now, according to the FBI, you now need a minimum of 20 markers before you can claim that a piece of evidence comes from a certain individual. This would not pass the test, not then and not now, to state that BZ is definitively from Teresa Habach. But if you look at the alleles that came up, one can see that it's highly suggestive of being Teresa Habach. And the reason why all the alleles did not come up was because the DNA, they used genomic DNA from tissue, it was too highly fragmented in order to get a result. If we have a look at Brendan Dassey, so we just had a look at the evidence against Stephen Avery. But if we have a look at Brendan Dassey, one thing that strikes out is a lack of any DNA evidence. And Sherry Culhane was questioned during the Stephen Avery trial. Question: did you ever find any DNA of a gentleman named Brendan Dassey anywhere in all of your tests? No, I did not. Question: not one shred, right? Answer: no, I did not find his DNA. Question: and you had his profile? Yes, I did. So there was actually no DNA evidence at all against Brendan Dassey. So let's have a look at the trial verdicts. As you have gathered, Stephen Avery has been sentenced to life in prison with no parole, and there was DNA evidence. Brendan Dassey has also been sent to life in prison, potential of parole in 2048, and there is no DNA evidence. It was all confession based. And just to let you know that Bron and I made a video, a YouTube video, that looked at Brendan's confession and whether his confession actually came out of the film Kiss the Girls. And you're invited to have a look if you haven't done so already. Okay, where do we go now? What's the status? As I said to you before, there is a box of cremains, human cremains. These cremains, the bones were extremely burnt and fragmented. They were recovered from multiple sites, including the Manitowoc gravel pit. And as yet, there's been no testing of any bone. The only thing that's been tested is a little bit of tissue. There is a new system known as the Andy Rapid DNA Testing System. This system allows you to obtain a DNA profile in about 90 minutes, so it's extremely quick. The other thing about the Andy system is that it's a very sensitive process. It's perfect for highly fragmented DNA molecules. Now, when the FBI did some analysis years back, they failed to obtain a DNA profile because they were using older, outdated methods. But now we have a new machine that should be able to test the bones. And again, as I stated in the beginning of my presentation, mitochondrial DNA is likely to have survived. Some mitochondrial DNA would likely have been able to survive, and it's still present in the bones. So it'll make an ideal test to determine whether a full DNA profile can be obtained. Essentially, it'll be tremendous to know whose bones they are and who they belong to. You may have heard, of course, of Kathleen Zellner, who's put in a lot of motions to the state, and so far the state is not allowing her or her experts to test the bones. And the question is, why won't the state allow the testing of the bones? In fact, in one of the motions, the state doesn't want any biological material to be tested. And furthermore, the state actually gave back many bones to the Habach family back in 2011. So there may not be any bones left to test. That's the end of my presentation. I hope that you've enjoyed it, and I hope that you've understood a little bit more about the biology in this case and the science behind the Steve Avery and Brendan Dassey cases.